So good afternoon all of you and welcome back to Excelsior Academy. So today we are going to do the next part of the cell cycle. So today we are going to learn about the S phase and the regulation of DNA replication. So in the previous lecture we completed about like what are the different phases of the cell cycle, what are the checkpoints and how it is being regulated at the G1 phase. So in this we are going to talk about the S phase and the regulation of DNA replication, since we know that DNA replication or DNA synthesis takes place in the S phase of the cell cycle. Yeah. Okay. So, so we know that CDK2 and cyclin E complex initiates the S phase by activating DNA synthesis at the replication origins. Okay. So remember this CDK2 and cyclin E. What about CDK4, 6 and cyclin D? That is of which phase?
G1, G1,
G1. Okay. And if it is CDK2 cyclin A, then which phase? If it is CDK2 and cyclin A, then which phase it is?
S2G,
Very good. That is G2. Yeah. Okay. So now mammalian cells use thousands of origins to replicate their DNA. Now one question, like for the interview, there was this question. I mean, these questions will be there for your MSc, after your MSc, but there is no harm in learning before itself. So the question was, in prokaryotes, there is one origin of replication, whereas in mammals or eukaryotes, you have got many. Okay. Now, why in case of mammals, we have got many? What will be the answer for this? It's a very simple answer. Yes, what will be the answer? Guess. It's okay, even if it is wrong, I'm there to help.
ma'am, repeat, ma'am.
So the question is that in prokaryotes, you have got one origin of replication, okay? Whereas in case of mammals or eukaryotic cell, you have got many origin of replication. Why this difference is there? Why in eukaryotes, many origin of replication is needed? Ori, it is called ori. Why so many are required? An answer is very simple. Any guesses, Pratima? It's simple. See, mammals means what? Eukaryotic cell means what? It has got huge amount of DNA. Large amount of DNA is there, right? And if in order to complete the replication, if suppose it was only one origin of replication, then it will take months and all to complete the replication. But you know that the S phase is hardly how many hours? 8 hours, I guess. In that 8 hours, you need to complete the entire DNA synthesis. And if you have got more chromosome, more time it will take for the DNA synthesis. That's why in order to reduce the time taken, you have got many origin of replication. Okay. Now the thing is that this origin of replication, even though many are there, but they are so tightly regulated that it will never repeat the replication within the same. It means what? Only in one cell cycle, you will have only one replication. Even though many are there, the cell doesn't get confused and starts the replication in few places and in places not. Okay, it's highly regulated. So it has been done in such a way that each origin of replication will start the replication only once per cell cycle. Okay. Now, the initiation of replication at each of these origins must be carefully controlled so that each segment of the genome is replicated once and only once, okay, during the S phase of the each cell cycle. So, same thing which I was mentioning here, that in one cell cycle, you will have only one replication of the DNA. Now, the DNA replication is initiated by the activity of MCM. Now, these are the protein terminology. Now you have to remember, okay? So mini chromosome maintenance, helicase. Now why helicase is required? In order to start the replication. So you know that DNA, it is there. So after that, we need, it has to open it, right? Then only in one direction, the other direction, your replication will start. And for this, we need helicase. Okay. So its complete name is mini chromosome maintenance helicase. which is again regulated by CDK cyclin complex at different stages of the cell cycle. So now today what we are going to learn in this particular session is origin of replication or the DNA replication, how it is being regulated. Okay. So the MCM helicase proteins will bind to replication origins together with the ORC. Okay, what is ORC? Origin recognition complex. So now what is happening? At your G1, you have got your origin of replication to which your ORC has already bound, and to this ORC MCM comes and binds. Okay, is this much clear? Over here the replication has not been started. It's just that the proteins go and bind to the origin of replication. Now what do you mean by origin of replication? Every time when I'm saying, it means there are certain sequences at which the replication will start. Okay. Okay, now this is the entire figure which we are going to discuss today. So you can see that this is your origin of replication. Okay, these are the sequences which is responsible to initiate the replication process, to which your ORC, what is ORC? Origin recognition complex. Okay, it will go and bind. Now once this complex is bound, to that your MCM will come and bind. Now MCM and ORC bound to the sequences meant for the replication is called pre-replication complex. Now this pre-replication complex is formed in your G1. Okay, don't get confused. We know that replication happens in the S phase, but the complex is formed in the G1 itself. Now, so they remain inactive as a pre-replication complex throughout G1 and becomes active only when it enters the S phase. Now, this activation of MCM in S phase results from the action of CDK2 cyclin E. So we know that CDK2 cyclin E is responsible for your S phase. Now, what is the function of CDK2 cyclin E in the S phase? So what it is doing is it is activating your MCM protein. Now, how it is activating? By phosphorylating it. Okay. Now, so activation of CDK2 cyclin E leads to inhibition of APC. This we have studied in the previous lecture also. And then what will happen? Since this gets inhibited, the level of cyclin E will increase, right? Now
Yes, ma'am.
that means the increase in cyclin E means increase in formation of CDK2 cyclin E. It means what? We have entered the S phase. Okay. Now, once your APC is being inhibited, the another protein kinase is there called DDK. Okay. Again, K stands for kinase. Now, DDK will go and phosphorylate your MCM protein. Okay. Now, what is happening? So once this is getting your phosphorylated. Now, so all your MCM protein, you can see that got phosphorylated. Now, this is giving the indication that the DNA strand needs to get open up. Okay. So your helicase activity will come into action. So the activation of the MCM helicase initiates DNA replication and the MCM proteins move away from the origin with the replication fork. So you can see that in the figure. Now it has been open. Okay, so it has unwind the DNA. Unwind the DNA. So this is called your replication fork also. Okay, a replication fork. So when we are going to talk about DNA replication in the mole bio section, it is the same figure, same proteins. Again, we are going to study. So that time it will be just like a revision for you guys. Okay. Now what is happening when the replication fork is formed? Obviously, your DNA polymerase are recruited to the replication fork and then they start the replication. Okay. So you know that one is going to be the leading strand and the other one is going to be your lagging strand. So this we know. Now, the high activity of CDKs during S phase, G2 and M phase prevents the MCM proteins from reassociating with the replication origin. Now, why this is important? Because we don't want to reinitiate the replication. That's the re-replication within the one cell cycle. It is not required. So that will be prevented. Okay. Thus, once an origin becomes active during S phase, replication at the origin cannot initiate again until the cell passes through the mitosis and enters the G1 phase of the next cell cycle. Okay, so far so good. Now you're able to understand how exactly the DNA replication is being it's regulated. Yeah.
Ma'am, just from once,
Okay.
from first one.
Yes, just see the figure. Okay, don't see the written part. Just see the figure now. Okay.
Yes, ma'am.
So you have got the DNA sequences, which is meant for your origin of replication. Origin of replication means what? From where the replication will start. Okay. To that, your ORC, origin recognition complex, goes and binds. Now, whenever ORC is there, it is going to recruit your helicase, and that is your MCM helicase. Now, this complex is formed. It is called pre-replication complex. It is found in the G1 phase of the cell cycle. Now, we know that CDK2 cyclin E concentration increases in the S phase of the cell cycle by inhibiting the activity of APC. Right? APC is your anaphase promoting complex. It is going to inhibit, it gets inhibited. Okay. Now, what will happen? The concentration of cyclin E will increase. It means CDK2 cyclin E complex will increase. Okay. Now, this is going to activate your DDK. This is going to activate your DDK. DDK is going to activate your MCM by phosphorylating it. So what will happen? The DNA sequence gets open up. Now once it is open up, this is called your replication fork. Okay. Now once this is being done, your DNA polymerase are recruited and in both the directions your DNA gets synthesized. So at the end of the S phase or in the G2 phase, you have got now two double-stranded DNA molecules, right? So it has been replicated.
Yes, ma'am.
Okay. Okay. Now, once we talked about the DNA replication, we need to know what are the DNA damage checkpoints. It means what? There are chances that during the cell cycle and the DNA replication, errors might have occurred. Either it is external or the internal. Now, when I say external factors which is going to affect the DNA or going to damage the DNA, it is we are talking about the gamma radiation or UV rays, that those are your physical damages. And if it is chemical, it means certain compounds will be there which can actually cleave your DNA, right? So in order to repair that, we need the checkpoints. So DNA damage checkpoints, cell proliferation is regulated not only by the growth factors but also by the signals that act to inhibit the cell cycle progression. So it is important to inhibit the cell progression in order to prevent becoming cancerous. Okay. So DNA damage checkpoints, they are very important, play important role also in maintaining the integrity of the genome by arresting the cell cycle progression. Now, they are operative in G1, S and G2 phases of the cell cycle. Now, if suppose these checkpoints are overcome, then what will happen? The cell will enter the M phase even if the DNA is not repaired. And after 2-3 generations, the cell will become cancerous. And that's how one cancer cell will give rise to many cancer cells, and you get the tumor and finally metastasis, and that becomes a problem for the individual to survive. Now, since suppose let us say that there is a DNA damage, that time your cell cycle arrest is mediated by two protein kinases. Again, remember, see, I always said that in cell cycle, all the proteins which we are going to study are kinases, okay? So these are known as ATR and ATM. The full forms are ataxia telangiecta rad3 and ataxia telangiecta mutated. So they get activated whenever there is a DNA damage response is being detected. Okay. So what are the two proteins getting activated? That is your ATR and ATM. Okay. Now, when they get activated, they give these signals not only for the activation of DNA repair, but also in some cases programmed cell death. So what is programmed cell death? It is apoptosis.
Apoptosis.
CD.
Yes. So this is your PCD1. Remember what is PCD2? I have asked this question. What is PCD2?
PCD2.
Autophagy.
Very good. It is autophagy. Okay. Now, The importance of this DNA damage response is emphasized by the fact that these proteins were initially identified because mutations to the gene coding AATM are responsible for the disease ataxia-telangiectasia, and that's why they have named like that those proteins. Okay, and finally, what happens? These people are having very poor immune system and have the high frequency of cancer. Okay, and that's why these proteins, when it was recognized in these patients, it was named ATM only. Okay, so ATM and ATR, they are the protein complexes that recognize the damage of the unreplicated DNA. So once they get activated, they will also have their consequences. Now, this is important to remember. Star. So ATR is activated by single-stranded or unreplicated DNA, while ATM is activated by double-stranded breaks. Okay. Now, you need to understand, you need to imagine actually, so how these proteins are working actually. See, you have got your DNA. Okay. So these proteins are actually, like this. They are like this. So entire DNA, double-stranded DNA, they will just go on scanning. So whenever any single-stranded DNA damage is there, so ATR will get activated. If they are scanning and they found that both the strands are broken, okay, and your DNA, double-stranded DNA here and double-stranded DNA is just hanging and there is a break over here. So what will happen? That time your ATM gets activated. Clear? Now, once activated by the DNA damage, ATR and ATM phosphorylate and activate the checkpoint kinase called CHK1 and CHK2 respectively. For ATR, it is CHK1. For ATM, it is CHK2. Okay. So what these checkpoint kinase are going to do? They are going to arrest the cell cycle by phosphorylating and inhibiting the degradation of CDC25 phosphatase. Now tell me, what was the action of CDC25 phosphatases?
It
Hmm?
inhibits
No.
dephosphorylating.
Yes, phosphatase means it is going to
Dephosphorylation.
dephosphorylate, but what? So by dephosphorylating, it is doing what?
It is making active
Yes, what?
MPF
Very
factor.
good, very good. So it is your CDK2, CDK1 and cyclin B, right? So they are having 3-phosphate group, remember, because of the V1 protein, protein kinase, and it is in the
Yes, ma'am. Yes.
right. So your CDC25 phosphatase goes and removes the phosphate group and makes your MPF active. Okay, then only it will go under the mitosis, right? Okay,
Mitosis.
good. So this is how your ATM and ATR is going to work. For single-stranded and unreplicated DNA, ATR is a which goes and just checks the DNA. So once it detects, suppose this damage is a check one, or that is your checkpoint kinase 1 will get activated. If it is a double-stranded break, ATM gets activated. So in both the cases, it is going to inhibit the activity of CDC25, whose action is the CDK2 and CDK1 activation. Okay. This is a figure in the previous lecture we talked about, right? So here, what is the function? You can see that CDC25 is there, which actually activates your MPF so that it can enter the mitosis. Now, once CDC25 is not activated, then the cell gets arrested into the G2 phase itself. It will not go into the mitosis. Okay. So CDC25 phosphatases are required to activate CDK2 1 and 2 by removing inhibitory phosphorylations during the cell cycle progression. DNA damage thus leads to inhibition of both CDK2, resulting in the cell cycle arrest in G1S, and CDK1 resulting in G2. Okay. So it is just the opposite, you need to remember. 2 means arresting at G1 and S. 1 means it is arresting at the G2 phase. That's all. Okay. Now this protein P53 is again very, very important. in point of with respect to the evolutionary significance also. So there is this article, if you get time and if interest is there, then you can study that. So P53 is there, okay? It is your tumor suppressor gene again. So this protein is actually present in double copy, not double copy, I think multiple copies in elephant. Okay, so in humans it is single copy, whereas in case of elephants they have got many multiple copies are there. Now why I'm discussing this is that because in case of P53, if it gets mutated, even a single small mutation is enough to disrupt its function. And what will happen if P53 gets mutated? It's a tumor suppressor protein. So what will happen?
It will get the tumors.
Yes, the person will get tumors. And it was observed that in the case of elephants, there are multiple copies. So even if mutation is happening and one and two copies are getting mutated, still there are other copies of the P53 which is able to do the function, right, of tumor suppressor. And because of this only, elephants are the only mammals which have never reported to have the cancer. Whereas in the other animals also it has been reported, but elephants are the only mammals in which till now cancers have not been reported. Okay, so that's how they are, correlating. Like it is only because of the multiple copies of P53 or something, or something else is also present. So that is also one way, to treat in future if we come to know what other proteins are involved. Anyways, okay, so coming to the P53. So in mammals, in mammalian cells, arresting a DNA damage checkpoint is also mediated by the action of P53. Now, P53 gets activated by both ATM and CHK2. Okay, so what is happening now? Phosphorylation stabilizes P53. So this is your P53 active, which is otherwise rapidly degraded. If it is not phosphorylated, then it is in the degraded form. Okay, so if it is phosphorylated, it means it is active. Okay, now who is going to activate the P53? ATM detects the double stranded break in the DNA. ATM is there, it goes and gets activates your CHK2. What is CHK2? Checkpoint 2. Correct. Now this gets activated, it is a kinase which will go and phosphorylate P53. So you can see that double phosphorylation is happening. Okay. Now once it gets phosphorylated, it is going to get increase in its level. So level is increasing. Now, P53 is a transcription factor and its increase in the level leads to the induction of CDK inhibitor P21, which inhibits the complex of CDK2 and cyclin or cyclin A. Remember CDK or P21 action? So we had CDK2 and we have cyclin E. So to that P21 was inhibiting. So it gets phosphorylated, then this has been removed with the help of the APC. If you remember the previous figure, are you able to remember the figure? Where we have discussed that the regulation is also with the help of the inhibitors, not only phosphorylation and this thing. Okay, no problem.
No.
Just go back and check again. Okay.
In the that is P27, no, ma'am.
Oh yes, P27. Sorry. Yeah, it is 27. Sorry, you have got the another also. CDK inhibitor P21 also. Okay.
Okay,
Yeah,
ma'am.
many other. Actually, many other. Yes. Sorry, my mistake. Many other actually. But the process is same. Okay. So it is going to inhibit the activity of CDK2 with E or A. So it means what? Where do we see E and where do we see A?
E is S phase and A is in G
G.
G2 phase.
So it means what? It is arresting the cell at that particular phase of the cell cycle. Okay. Now, what happens if you lose the activity of P53? Obviously, cancer. See, whenever we talk about cell bio, anything which is beyond the normal, it means it is going to form the cancer. Okay. So, after learning for so many years and doing experiments, learning, being at Tata Cancer Hospital also, I have understood that there is no treatment for cancer. Yeah, And really, I mean, you can just delay the growth of the cancer, but it is very difficult to treat. I mean, if you say that this person is free of cancer, I mean, after 5 or 10 years, there is 95% chance that that person is again going to get the cancer. So it's very difficult. Okay. So loss of P53 function as a result of these mutation will prevent the cell cycle damage, right? So what will happen? It will be passed, the damaged DNA will be passed on to the daughter cells, which for which the DNA repair is not possible. Now, since it is not possible, what will happen? The mutation starts getting accumulated, and for a cell to become a for a cell to become a cancer, minimum of 4 to 5 mutations are required. So when these mutations or the damaged DNA gets all accumulated, eventually the cell becomes cancer. Okay. So everything is going to contribute for the cancer development. So mutation in the P53 gene are among the most common genetic alterations in human cancers, illustrating the critical importance of cell cycle regulation in the life of multicellular organism. So this sentence says that let it be any type of cancer, you will always see that P53 has mutated because it's a part of regulating your cell cycle. And in the tumor and in the cancer, we know that the cell cycle is not under the normal phase, right? It has shown the uncontrolled growth. So obviously it can be any kind of cancer, and there will be P53 mutation already happened. Okay, so that's all about today, the about the S phase and the cell cycle. That is the DNA replication part, and what is left is about the mitosis. Okay, and after that your what to say, the cell cycle gets completed, and then our topic what we'll have is cell signaling. Okay, yeah. So thank you guys.
Okay, ma'am.
Any doubts?